So FACS full form is Fluorescence Activated Cell Sorting uh, Technique. So FACS. Uh, so we will go through. It's mainly what we'll do. We differentiate between the cell type, how the cell looks like, its shape, and how the cell morphology is. Is it a complex or simple? We can differentiate based on that in this case. Um, so whether the cell is small or whether the cell is big, like white blood cells, red blood cells, leukocytes, basomocytes. So all the cells have different sizes. So that's what the blood test samples are have been taken from you, and they are put them into the flow cytometer to check your blood count, your RBC, how many RBCs are present, how many WCs are present, white blood cells. So they all being sorted out with the help of this technique based on their morphology and size. So basically, what you do is your cell just go through um, an analyzer of the FACS analysis, and it's been struck by a laser light. And then after the striking the laser light, basically your cell gives two scatters. One is forward scattered. One is uh, yeah, side scattered. Forward scattered was there, and something was side scattered. It's scattered on the other sides. So one will be forwardly scattered, one will be on the sideways scattered, and then based on that you have two values, and you make a 2D histogram, and that 2D histogram you put the values, and you can check with the thing how things are. So this was done. This is simple cell sorting. In the fluorescence activated sorting, you put some fluorescence to your cells. So when they are activated, so they give different fluorescence. If you have two different cell types like CD14, CD8, two different cell types, and you want to have different fluorescence coming up, so you can check with the help of fluorescence activated cell sorting machine, FACS machine, with the different colors coming up, and then you will have different fluorescence for different cells. So based on that cell type versus fluorescence, so you can make 2D histogram of that also, and you can have differentiated results. So that's the main summary of FACS analysis for today. So we will go through now in more details about it. Let's start. So in this topic, mainly we will deal with the basics of flow cytometry, understand how flow cytometry sort of works, understand how cells are sorted out, description of different cell sorting possibilities, tips on sample preparations, overview of sorting service provided by FCS. So this is done by some BD Biosciences. Uh, so so these techniques are shown you live, which techniques are appropriate for my cell separation. So if you have a sample, either it will be in tissue or it will be in the suspension form in fluid. If it is in tissue, then you go for laser micro dissection microscopy. If it is in the fluid form, then you can go for FACS analysis or microfluidic techniques like depth array, Tito array, or large display sortings like COPAS. So these are the basic three other techniques for the same. So what is flow cytometry? How's the name came from? Flow means fluid, cyto means cells, metry means measurement. So measurement of fluid cells. The cells which are in fluid, to measure them is known as flow cytometry. So what type of particles can be analyzed? Basically, cells, chromosomes, bacteria, yeast, microparticles, all these uh, these particles, organisms, cellular structures are possible to measure of different size. Actually, you can see. So particle size that can be analyzed from 0.5 micrometer to 14 micrometer. So you can see red blood cells being 6 micrometer, lymphocytes 8 micrometer, neutrophil 12 micrometer, monocytes 14 micrometer. So you can sort all these cells according to the size, according to the structure, according to the morphology, smallest to the largest. So what does it matter? Three key parameters are measured in this: relative size, relative internal complexity, and fluorescence intensity. So if it is simple sorting, then the first two things will be checked: the size and the complexity, the morphology of the cells. Otherwise, if your fluorescence is there, then relative fluorescence would be added in the FACS analysis. Then these are the various samples that are being run. These are the various analyzers. Uh, some are from they are mostly from BD Bioscience, BD FACS, various companies names are there. You can check them out. So so they help in the forward and side scattering, both way of doing it. They're helping out. So each has its own ease and easy to go. So here, like if you put your samples in the tubes here one sample is possible at once. Here multiple samples are possible. This is the most, I think, mobile machines is possible for these lower somewhere where it's not so much FACS is being used at a lower level they want to go for. So each one has its own applications according to their. So sorter can be divided into three main subsystems, that is fluidics, optics, and electronics. So sorter can be divided into three main subsystems: fluidics, optics, electronics, to introduce and focus the cells for interrogation and create a stable break off for sorting. Optics to generate the collect and light signals, and electronics to convert your data into digital signals and process the signals and communicate with the computer. So these are the three main things. So your cells are mainly being recorded into this hydrodynamically focused in the core stream like this one. So you have a central core, you have a sheet fluid coming up, and a single flow is going on. And your sheet flow and the central core, they are not being mixed; they are being separated. So sample is introduced into the running sheet fluid in the flow cell, then sample in the hydrodynamical focus in the core stream. Then sheet fluid samples do not mix. Then reduction of cross section causes the acceleration of central core team leads to a laminar fluidex. Then sample flow rate adjustable. And later on the this rate is acceptable. Then its speed minimum is 10 microliter per minute, medium is 35, highest is the 60 microliter per minute. So there are two different optic systems in sorter. So one is excitation optics consisting of lasers and fiber optic cables. So these are fiber optics here, and then there's a X-mount optical plates. Then prism is there. Then lenses are there. Then flow cell is going on with the nozzle here. So various laser lights, they're coming from the fiber optics, and with the help of prism being focused, and then with the lenses even more focused, then your laser lights are going like this. So it's also consisting of fiber optic cables that direct the emitted light to the appropriate emission, and filters that direct the signals into the emission to the appropriate photon multiplier tube, PMT. So with the help of PMT, appropriately where it has to go, it directly send it to the concerned place. So these are your various arrangements of your adapters. So this is trigon and octagon, so A, B, C, and here starting from A, then B, then C, then D, E, F, G. So mainly sort of they try to make it in opposite direction, these adapters. Then PMT looks like this, where you have a photon of light coming out from there, from the cathode, electron beam is gone. Then there are diodes from where it's getting heated very widely in the vacuum tube, and then it goes to anode. Then it could be get through the currents via this. Current could be seen from this. So how cells are sorted? This is just basic telling you like you have a stream of uh, your cell are being sorted from here. One would be one will be the positive side, one will be the negative side, and in the center will be the neutral. So they will be deflected as per the things are. So once your cells are dropping in the charge droplets, so like this. So first of all, sample generates light scatter and fluorescent signals. Signals are analyzed. So there is a fluorescent and light scattered signals are being generated. Then charge is applied by a stream charging wire. Then charge droplets break off because of that here. Then deflection plates attract or repel charge droplets. One will be positive side, one will be negative, and then they will be discarded according to their charges. Uncharged dot pass to the waste, which are the uncharged, they will go into the waste in the center, and the charged droplets containing particles of interest are collected here. So, like in this case, so maybe red ones and green ones. If we have fluorescently sorted our cells, so they will be separated like that. So this is more into the practical work. If you want to go to know about it, how it is done in practical. So this is how the droplets looks like. So there's a break off is happening, then there's being sorted out, being green fluorescent and orange fluorescent, and the center is the neutral one from here. And this is the breakup point from where they started to separate it, and on top is interrogation points. You can see how the droplet is going on like this. So they're just telling that how with the different methods, how you can take care of the droplets. Sometimes it has long droplets, which is not good. It's better to have small droplets so that the results could be better. So that's it's all about telling. So I will not go in details. It's not required for us. So I will just, because we are learning the basic principle, um, and it's quite, it will not make sense to go in details so far with this. So into what collection devices can I sort my samples? You can use 6 well plate, you can use 8 well plate, you can use 12 well plate, you can use 96 well plate, you can use 364 well plates, you can use any well plate as per you are looking for samples. So the bulk sorts are done with the purpose of enrichment of cell culture for like DNA, RNA analysis, adoptive transfer, purification of cells expressing fluorescent protein. So cell could be sorted into 1.5 ml per tubes, 4 population parallel, 5 ml FACS tube, 15 ml tubes. So these are all things are possible. So it could also be sorted from the 6 to 384 well plate if you want to. So these are various population sorters using different experiments. So plan your experiments, choose your fluorescent antibodies when you're gonna do your experiments, choose wisely when you're doing experiments, bring along the following controls, unstained or to estimate the background signals, so the one which are non-transfected, unstained, bring single strain samples to set compensation, and bring FMOs if needs to set gates. Maximum of 2% of FCS fetal calf serum or BSA is recommended in the suspension medium because they will attach to the cells. So avoid phenol red media due to autofluorescence. They could get fluorescence, which is false. Then it's better to cells do well with the PBS or FCS and BSA. And if your cells have tendency to aggregate, consider adding one of the following: calcium or magnesium in free sorting buffer, or add EDTA. So these are the things, bachcho, you have to take care, students, when you are gonna start your experiments with the FACS analysis, that to get the results to be better, there should be no background noise, no data, false readouts. Everything that you're doing is done right. And all samples need to be filtered, preferably directly before sorting. For this, for bulk sort, provide cells with the following concentration depending on the used nozzle. So if you're using a nozzle of 770 a micrometer nozzle, then your cell should be 40 to 10 raised to over 6 cells per ml. So as per the nozzle, your cells should be there, concentration should be present. Otherwise, it will be also not good. That cells will be not coming nicely from the nozzle then. So that's it for us, I guess. That's a summary what we did. Thank you so much from the Edufabrica team that you had this nice workshop. Now I will take you to the excited part of facts analysis, where you will learn whatever I have told you, that you can differentiate by forward scattered and side scattered thingy, and also with the fluorescence. Now we will learn them with the nice diagrams, the nice pictures, and this nice animation video.
For a very interesting mechanism called the flow cytometer and FACS. Actually, I did 2 videos, so in this video I'm going to mention specifically flow cytometer, and in the next video I'm going to mention the FACS mechanism, fluorescence activated cell sorting. Let's start speaking about flow cytometer. Flow cytometer is a cell counting mechanism. So it's used to count the cells and to analyze the phenotype and the health situation of the cell. How does it work in general? First, in general, in flow cytometer, I have something called the flow cell. In the flow cell, we add our cell mixture. Then I have the sheath fluids. The sheath fluids will apply something called the hydrodynamic focusing. The hydrodynamic focusing of the sheath fluids will send the cells one by one through the flow cytometer tube. It's very important to pass the cells one by one through the tube, because here I have something called the laser beam, and the cells should pass through the laser beam one by one. This point where the cells pass through the laser beam is called the interrogation point. Now, when the cell pass through the laser beam, there are 2 events happening. First, it's the forward scattering, which is the light scattered in an acute angle to the forward direction, and the side scattering, which is the light scattered in every other direction, in side directions. These 2 events, or this light scattering, is detected by specific detectors called the forward detector and the side detectors. And these detectors are connected to a PC or a data analyzer, which is going to analyze the data obtained from the detectors. Now, this is in general. Now, I'm going to speak more in details. Saying that this is the flow cytometer tube, and then the cells pass one by one through the laser beam. When the cell pass through the laser beam, the cells scatter the light in a forward direction, which is the light scattered in an acute angle. This forward scattered light is directly proportional to the size of the cell. Why? Because the cells scatter as like a particular amount of light in the forward direction, but when the cell is bigger, the forward scattered light will be larger, or like more forward scattered light will be detected. And when the cell is smaller, the cell will scatter less light in the forward direction. So the forward scattered light is directly proportional to the size of the cell. Now, the detector detects the pulse of photons, which are scattered light is a pulse of photons which are detected by the detector. And the detector then is, as I told you before, is connected to a PC or a computer. The PC will convert the intensity of the light detected by the detector into voltage. And then I will get a specific amount of voltage for every cell passing through the laser beam. The PC will give me a data similar to this. So every time a cell passes through the laser beam, I will get a specific peak like high voltage peak for large cells and low voltage peaks for small cells. The data or the forward scattered light data at the end will be similar to this. Time versus voltage, and then the peak will be proportional to the size of the cell. This is forward scattered light. The second event happening in the flow cytometer is the side scattered light. So when a cell passes through the laser beam, the cell will scatter the light in side direction. And this side scattered light is directly proportional to the complexity and granularity of the cell. Why? Because the simple cells scatter the light in the side direction. Yes, but when we have granules inside the cell, so when the cell is complex from inside, these little granules will also scatter the light in different directions, and in this case I will get more side scattered light, like this. So this is a complex cell from inside. It contains small granules inside, and then these granules will also scatter the light in different directions, and in this case I will get higher intensity of the side scattered light. Similarly to what we saw in forward scattered light, the detectors are connected to the PC, which is going to convert the intensity of the light into voltage, and then I'll get something similar to this. So the side scattered light data will be similar to the forward scattered light data. So for the complex cells will give me a high peak, and the simple cells will give me a small peak. Now, in order to analyze the cells, what I have to do is that I can combine both forward scattered light and side scattered light data together. In this case, I'll get something called the two-dimensional histogram. The two-dimensional histogram, from its name, it's two-dimensional. So I have side scatter and forward scatter data all together. In this case, what I get is something similar to this. So I have here 4 quadrants. If the cell appears in the lower quadrants, it means that it has low signal on the side scatter detectors, which means that the cell is simple. If the cell appears in the upper half, it means that it gives high signal on the side scatter detectors, so it means that the cell is complex. If the cell appears on the right half, it means that the cell gives high signal on the forward scatter, which means that the cell is big. And if the cell appears here, it means that the cell is small. So if the cell appears, for example, in this quadrant, it means that the cell is small and it's simple. If the cell appears here, it means that the cell is small but it's complex. Here, it means that the cell is big and complex, and here, it means that the cell is big but simple. I will give you a very good example of this, which I have an example about the analysis of a sample of white blood cells. So what we see here is an analysis of white blood cells. This is side scatter and this is forward scatter light. What we see here is a group of cells which are complex, very complex cells, because they give high signal on the side scatter detector, and they are medium inside. They are not so big and they are not small, so they are medium inside. Obviously, these cells are granulocytes, because granulocytes, or like eosinophil, basophil, neutrophils, they are medium inside, but they are very complex cells. Here we have bigger cells, because they give almost like they give more or less high signal on the forward scatter, but they give low signal on the side scatter. So they are big, but they are not complex. They are simple cells, and these are monocytes, because monocytes are big cells, but they are not complex. Here we have very simple cells because they give very low signal on the side scatter, and they are like smaller than monocytes and granulocytes. So obviously these are lymphocytes because the lymphocytes are smaller in size and they are simple cells. Of course, this is a very useful technique used to count the cells and to analyze the phenotype and the health situation of the cells. And what's very special about this technique is that we can analyze many thousands of particles and cells per second. It's like it's an easy technique. It's not very hard to perform, and it's precise. It's very used nowadays in all the laboratories. This is everything I wanted to tell you about flow cytometer, which is very important to know if you want to know more about FACS mechanism. In the next video, I will speak about FACS mechanism. So if you want to know more about FACS mechanism, then you need to watch.
Okay,
Hello guys, this is a continuation of the subject flow cytometer and FACS mechanism. In the previous video, I spoke in details about flow cytometer, and in this video I'm going to speak about FACS mechanism, fluorescence activated cell sorting. If you didn't watch the previous video, I highly recommend you to go and watch it, because it will be much easier to understand FACS mechanism if you fully understand the flow cytometer mechanism. So let's start speaking about FACS. FACS mechanism is not only a cell counting mechanism, but it's also a cell sorting mechanism. So it provides a method to sort a heterogeneous mixture of biological cells. It depends on light scattering, as we saw in flow cytometer, so forward scattering in light and side scattering light, but it also depends on the fluorescence, and it also can detect fluorescence. FACS mechanism looks like this. So we have the machine, we have of course the computer, which is going to analyze the data, and we have the fluids container and the waste container. Now, how does it work? A FACS machine looks like this from inside. So we have also the flow cell and the sheath fluids, which apply the hydrodynamic forces on the cell to pass them one by one through the tube. This is what we saw in flow cytometer in the previous video. The cells will pass one by one through the cell tube, and then we have the laser. So the cells will pass through the laser beam one by one. When the cell pass through the laser beam, there is the forward scatter, which is detected by the forward scatter detector, and then there is the side scatter, which is detected by the side scatter detector. But here there is also the fluorescence, which is detected by the fluorescence detectors. Here we have mirrors and filters, which are aimed to filter the light and to deliver particular wavelengths to the appropriate detector. Then all the detectors are connected to the computer, which is going to analyze the data. Now let's speak more in details. As we saw in in flow cytometer, we have forward scatter and light and side scatter. Here we have also the fluorescence. So saying that I have a cell mixture. Inside this mixture, I have a mixture of cells, or cells who are expressing a particular protein and other cells who are not expressing the particular proteins, the particular protein or the protein of interest. What I want to do here is I want to count how many cells there is inside the mixture and how many cells are expressing the protein and how many cells are not expressing the protein. And then I want to separate this heterogeneous mixture into two groups of cells. The cells who are expressing the protein and the cells who are not expressing the protein. To do this, first of all, I have to design a specific antibody for this protein. This antibody is specific to the protein of my interest. I'm not like I can buy this protein. There are companies who design specific antibodies for particular proteins. Then I have to label this antibody with a specific fluorophore. Now what I have to do is that I have to add this antibody, labeled antibody, to the mixture of cells I have. When the antibody is added to the cells, this antibody is going to bind specifically to the protein of my interest, and then I will have labeled cells. So I will have the cells who are expressing the protein will be labeled with this fluorophore. Now, when the cell passes through the laser beam, this is going to happen. First, I will get the forward scattered light, which is detected by the forward scatter detector, and this is proportional to the size, as we saw in the previous. We saw this in details in the previous video. So forward scattered light is directly proportional to the size of the cell. Side scattered light is directly proportional to the complexity of the cell. And then the fluorescence will give me an information about which one of the cells is expressing the protein and which one of the cells is not expressing the protein. Now when the cell pass through the laser beam, this is the laser beam. First of all, this is the forward scattered, which is detected by the forward scatter detector. The side scatter, which is detected by the side scatter detectors. And then when the fluorophore passes through the laser beam, the fluorophore will be excited. And then when the fluorophore is excited, the fluorophore will emit light at a particular wavelength, and then this light will be detected by the fluorophore detectors. All these detectors are connected to the computer, which is going to give me the exact data about every event of these three. Now, of course, when the cell is expressing more proteins, there will be more fluorescence coming out of this cell. At the end, I will get a data similar to this. So when the cell is expressing the protein, I will get a high peak, and then when the cell is not expressing the protein, I will get a small peak. And then the computer will give me something called the one-dimensional histogram, which is something similar to this. Here we have the cell count, or the events. We call them the events passing through the laser beam. And here we have the signal of the fluorophore. So when I have a high signal of fluorophore, then these cells are the cells who are expressing the protein of interest, and the cells who give low signal on the fluorescence detectors are the cells who are not expressing the protein. What I can do further is to combine this data with other data I have. So for example, I can combine the data obtained from the fluorescence detectors with the data obtained from the forward scatter detectors, for example. So here I have the fluorophore, here I have the forward scatter, or here I have the fluorescence and forward scatter light. When the cell appears here, it means that these cells are small cells because they have low signal on the forward scatter and they have low signal on the fluorescence detectors, so they are not expressing the protein. When the cell appears here, it means that the cell is not expressing the protein because it has low fluorescence, but it's a big cell because it has high signal on the forward scatter light. Then when the cell appears here, it means that it's expressing the protein, but it's a small cell. And when the cell appears on the upper right quadrant, it means that the cell is big and it's expressing the protein. So this is what we call the two-dimensional histogram. I can combine two data together and then I get something like this. Now, what's special about FACS mechanism, or why it's called FACS? FACS means fluorescence activated cell sorting, which means that this mechanism can not only give me a cell count, cannot give me a data about the cell count or the percentage of the cell expressing the protein and non-expressing the protein, but also the FACS can separate the cell mixture into two groups of cells. How does it work? Let's see. This is the FACS mechanism. So here we have the flow cell, and here we have the cells passing one by one through the laser beam. When the cells pass one by one through the laser beam, as we saw before, it will scatter light forward and side scatter light, and also fluorescence. All these events will be detected by the detector. At the end of the tube, there is something called drop formation. In drop formation, we have a mechanism called a vibrating mechanism. This vibrating mechanism will separate my mixture into small droplets. These droplets should contain only one cell by droplet. This is very important. Then what I can do as a user is that I can adjust the settings of the program in order to charge the cells. So at the end of the tube, I have something called the electrical charging ring. The electrical charging ring is responsible to charge the cells according to my adjustment. So, for example, if I adjust the settings, for example, of the program, I can ask the computer to charge the high fluorescence cells with a positive charge and the low fluorescence cells with a negative charge. Then the electrical charging ring will charge the cells will pass through the laser beam. The detector will detect the fluorescence, and then the computer will charge the high fluorescent cell with positive charge, for example, and the low fluorescent cells with negative charge. I can choose, actually. For example, if I have this data, this is the two-dimensional histogram, I can choose to charge the big high fluorescent cells with a positive charge, and the big low fluorescent cells with a negative charge, and the other cells with no charge. Then the neutral cells will go to the waste. These cells might be like debris. These might be like just debris. I'm not interested in them. So it depends what cells I'm interested in. So I can adjust the settings of the program, and then the electrical charging ring will charge the cells. Then I have different electrodes, like negative and positive electrodes, and then the positive cells will go to the negative electrodes, and the negative cells will go to the positive electrodes, and the neutral cells will go to the waste. In this case, I will have two groups of cells. The cells who are expressing the protein and the cells who are not expressing the protein. And this is what we call cell sorting. Of course, I can also study two proteins, not only one. And this is done by using different... of course, to study two proteins, I need two specific antibodies, because I need one specific antibody for each protein. But I need to label every antibody with a different fluorophore. Of course, I have different fluorophores, and every fluorophore emits light in a different wavelength. I need to know every fluorophore, the wavelength every fluorophore emits light in. So if I use two different fluorophores, then I can study two different proteins. For example, this fluorophore emits a green light or a light green light, and this fluorophore emits yellow light. So they are different wavelengths. Then if the cell is emitting only has only a green fluorophore, we can know that this cell is expressing one protein, or the green protein. If the cell is emitting yellow light, then the cell is expressing only the second protein. And if the cell is emitting two lights, or giving two signals on the two detectors, then we can say that the cell is expressing both proteins. How can we detect two different wavelengths? As I showed you before, The FACS mechanism contains different detectors, different fluorescence detectors, and here we have the filters, the light filters, which deliver a particular wavelength to a particular detector. So every detector we detect a particular wavelength, and then we can use two different fluorophores if they emit light on different wavelengths. Okay. We can also combine the data together. So I can form this two-dimensional histogram again, but with protein 1 and protein 2. So I have fluorophore 1 signal coming from fluorophore 1 and fluorophore 2. So when the cells are in this, when the cells are here, it means that these cells are expressing both proteins because they are giving high signal on the first detector on the second detector. If the cells are here, it means that they are expressing protein 2, but they are not expressing protein 1. These cells are expressing protein 1 because they have high signal on the detector of fluorophore 1, but they are not expressing protein 2 because they have low signal on the detector of fluorophore 2. And these cells are not expressing any of the proteins. Now I have like a real two-dimensional histogram. And this is like ideal to see how cells are shown in the two-dimensional histogram. So these cells, here we are studying two different proteins, CD14 and CD4. We are labeling CD14 with PE, and we are labeling CD4 with FET-C. We are labeling them with different fluorophores. Then cells in this quadrum are expressing CD14, but they are not expressing CD4, so we have CD4 negative. Cells here are expressing both. They are giving high signal on both of them. So the CD14 positive, CD4 positive. These cells are CD14 and CD4 negative because they are giving low signal on both detectors. And these cells are expressing CD4. They are giving high signal on FET-C detector, but they are giving low signal on the other detectors, so they are expressing only CD4. This is like a very simple example about how the data looks like in the two-dimensional histogram. Of course, there are many more complex data. So if you want me, I cannot speak about everything in this video. So if you want me to do another video especially about data analysis and gating in FACS analysis, please write me in the comments. As you know, now we can in the new FACS machines, we can analyze like tens of proteins and tens of cells in one time, because new FACS machines have like some machines like there is now one FACS machine in the market which contains 10 lasers and 30 detectors. Can you imagine 10 lasers and 30 detectors? So you can analyze many proteins and many cells at one time. But of course, the data then will be much more complex than this data. This is a very simple data. If you want me to talk more about this, then write me in the comments. If you enjoyed the video, don't hesitate to like the video, share, subscribe to the channel.
Okay. I hope she had cleared our concept quite clear. So what we did in the presentation, things are better than before, and we are able to understand things now, how the facts analysis is done. So there is one last video. This one. This shows the same thing what we have understand in 2D. Now we will understand in 3D. So I just accelerating the fun part today with these videos.
In this animation, to the flow cytometry experimental technique. Flow cytometry is used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific flow cytometry software programs. Once a suspension of individual cells has been prepared for flow cytometry analysis from cells, tissues, or organisms, the prepared sample is placed on the flow cytometer. How does a flow cytometer analyze the size, shape, and properties of individual cells? A flow cytometer contains several key components, including the sample, fluidics that move the sample into the flow cytometer, lasers, optics which gather the light, detectors to sense the light, and a computer system to output the data into a form that can be analyzed by the researcher. Once a sample has been placed in the flow cytometer, the machine will suck up the sample, mix the sample into a stream of saline solution in the cytometer, and lead the cell suspension through a narrowing channel, causing the cells to form a single file line before the cells pass through the laser at the interrogation point. Each cell within the sample then moves across the laser beam, allowing each cell within the sample to be individually analyzed. As each cell passes through the laser beam will scatter in multiple directions. The flow cytometer detects light scattered in a forward manner, called forward scatter, and light scattered in a sideways manner, called side scatter. The amount of forward scattered light for each cell is detected by a detector on the far side of the cell from the laser. Forward scatter is proportional to the size of the cell. The detector converts the scattered light into a voltage pulse, which is directly proportional to the amount of forward scattered light. The computer converts these data into a histogram plot with the amount of forward scattered light on the x-axis and the number of cells on the y-axis. The amount of side scattered light is detected by a detector located perpendicular to the path of the laser beam. Side scatter is proportional to the shape and internal complexity of a cell. Just like with forward scattered light, the flow cytometer converts the detected side scattered light into a voltage pulse, which is directly proportional to the amount of side scattered light. By analyzing the forward and side scatter data together, the researcher can understand a cell's size, shape, and complexity. In addition, the analysis of the forward and side scatter data can allow the researcher to divide the heterogeneous population of cells into individual populations with varying size, shape, and complexity. This capability of analyzing multiple populations within a sample is the immense strength of the flow cytometry experimental technique. In addition to analyzing a cell's size, shape, and complexity, a flow cytometer can also detect emitted light from excited fluorescent molecules, such as fluorescently labeled antibodies or fluorescent dyes or stains. Animation, we will focus on the detection of fluorescent signal by the flow cytometer. For more information about fluorescence, please see the fluorescence animation. The fluorescent molecules, or fluorophores, are excited by the correct wavelength of laser light when the cell passes through the laser beam. After excitation, the light emitted by the fluorophore is directed along a path with emission filters that allow for the detection of fluorophores emitting light in a cell. A flow cytometer can detect the presence of a fluorophore and also quantify the relative amount of a fluorophore within a cell. A flow cytometer can also be used to detect light emitted from multiple fluorophores in the same sample. While it is common for flow cytometers to detect 2, 3, or 4 colors at the same time, some flow cytometers can even detect up to 18 different colors at the same time. The data can then be presented to the researcher in the form of a histogram or a dot plot. Such data furthers the powers of flow cytometry analysis by enabling the researcher to analyze individual populations of cells within a heterogeneous mix of cells in the sample.
Okay. So class, that was facts analysis in detailed manner. So today there will be no 5 minutes break because we are done for today. I have to teach you about facts analysis today, and we did it in quite detailed manner. So main concept is clear between two types, one with the sorting and one with the fluorescence, how you can do it. So if you have any questions, you can ask now. Uh, so today will be 1 hour session. Anything you want to add? Intracellular proteins. Yes, yes. My friend did on the stem cell culture. She was creating a lot of human stem cell culture, and she was doing a lot of FACS analysis with them. So she was working on the proteins itself. Uh, okay. Mr. Moin, just write me in personal chat into this telegram. We can talk about this Ramachandra plot. This is a very basic biochemistry technique actually, and I had some slides for that, I guess, so I can share with you later. I can share with you later. Yeah, today my voice is not with me, but I hope you understand the data. Everything is sorted out. Uh, बच्चे sites for publication. Let me check. So Journal of Molecular Biology, I think it's free. Molecular Biology Journal, journal ranking on molecular biology. So Nature, molecular cell metabolism. Just so there are around 50 journals. Just go through them. Maybe I can share link on the YouTube. Just go through all these journals. These are molecular biology journals, and just see if you got luck with various impact fact papers and all details. Just check on the website to get the license, but mostly it's you just skip that. It will be working for you, but you have to buy it actually to get the license. There are other ways also to jailbreak kind of thing. I will not recommend that. Just buy the original one. So yes, you can leave. We are done for today. Today was 1 hour session. We have to do this on the 8-day facts analysis. We had a quite things done, and yeah. So good night. Take care. We see us tomorrow then. Either for the bioinformatics we will go or for the applications. Let's see. Uh, whatever the things is best for us. Okay, great. Have a nice night and sleep well. Stay safe. Take care. See you tomorrow then. I will end the stream now.